would like to thank you for the Aquaculture and Fisheries Congress Tokyo 2023 committee who allow me to our talk be present in this occasion. So our talk will be settlement of the settlement of the tropical box mussel Septifer bilocularis, effect of site, position, and substratum. So we are 4 persons here. 3 of us, me, Vanes Boneka, Erika Legis, come from the same university, which is Samratulangi University. And then we have Stella Kaunang, who come from Catholic University de La Salle, Manado. So can you see the next slide?
Yes, Medy. Yeah.
Okay, thank you. So my talk will be divided into introductions. I have methods and some results here, and then finally will be conclusions. Can you see now?
Yes, Medy, it's visible.
Okay, my organism is called Septifer bilocularis, which is known as box tropical mussels. Usually, these mussels attached at the bottom perform the aggregations. So the aggregations is not can be habitat for benthos, not only flora but also fauna. You can see at the right side of me, at the top of the picture, I have an example of our tropical box mussel. But then 2 pictures after that, we have an example of aggregation, which is the species is not a tropical mussel, but is one is Mytilus edulis from Europe. But this is only example the aggregation performing. So usually the mussels, they as known as filter feeders. When they are in the group aggregation, they can filter the water column, especially particle as they food, up to several meters from the sea bottom, actually. So mostly mussels, they produce larvae who stay at the water for some times, but then at the end of larva stage, they will go down at the bottom, swim, crawl, and if they to find the good substrate. If they find a good substrate, and then they will produce a byssus, they will attach to the substrate. So the good substrates, it depends. I think we know that whether physical, biological, or chemical properties actually. But then when they find the good substrate, then the larva behavior will be important one. But the other things also, the environment factors like what is called currents, especially when in the intertidal, it's quite important we consider about the current of when low water level comes and high water level comes. So in the land, we have different materials, such as we call palm, coconut, or plastic like polypropylene fibers that usually used by men to different purposes. But then it's also transferred to the sea by fishermen. They form like different kind of rope that can be used for different purposes too. But then when they are not used, so they sank at the bottom and become a junk or garbage. So this material actually, we are looking to find out whether our box mussel larva attached on it. With other materials, we use which called mussel cells with base substrate. So actually, we try to find out what is called whether there is any effect of sites, because we have different sites or stations. What is called position impacts, and also the effect, if any effect of the what is called substratum. So here at the right side, we have what's called North Sulawesi Islands, and we have two red cycle. At the northwest is called Tioho as one side, but then southwest, another cycle we call Blongo. So that's our two sites actually that we run the experiment. But then we have at the left side, we have the Indonesian islands. We have 5 big islands, but then we have the Sulawesi Islands that you can see a shape like a K. So, and then we are on the top north of Sulawesi Island actually, with the red square. I think you can see it. So I move to the, so here on the right side actually, I should mention again, we have the box mussel, but then down there we have large aggregations. So here in related to our experiment, we defined like edge of large aggregation, and then we have a middle of large aggregations. But then we have also which is called small aggregations. For the large aggregation, usually we have the diameter about 2 meters. But then we have a small aggregations, actually less than a half meter. But then at the live side, we have 20 times 20 square. Actually is polyvinyl chloride, PVC. So in this PVC plate, we make 16 hole, where each of the hole we have 1.2 centimeters. But then so the totally of the hole here we have 16, yeah. But then on the left side we have which the substrate. One is called palm fiber, And another one we call coconut fiber is brief with the CF. But then we have polypropylene fibers we call PPV. And the last substrate we call mussel cell with bisubstrate, MSB, we call that one. But then each of these substrate, 4 types of substrate, we have 4 replicates. That's mean totally 4 substrate, each with 4 replicates, we have 16. And this substrate we place in this 16 hole randomly. Yeah. But then, okay, I explained already the aggregation that actually we choose arbitrarily 3 large aggregation and another 3 different small aggregation. Again, actually we choose 3 different large aggregations, and also we choose 3 different small aggregations. The large aggregation has size diameter up to 2 meters. The small aggregation has size around less than half meters. So actually, each large aggregation with edge, actually we put one PVC plate at edge at the middle of large parts. And then we also put at the small aggregation with one PVC plate. But then we have another 2 more large aggregations. So also at each of edge and middle of the large, we also put the same PVC plate which already fit with a different kind of the substrate, as well as another 2 small aggregation. We put also each one PVC plate. So I hope you can get it. Now is the result. If you see this picture, so the larva, when they attach to the substrate, they not only attach to the fiber surfaces, but also they attach to among the fibers, whether palm or coconut or plastic fibers, and usually on the surface or in between the fibers. But then we have another ones, they attach on the cell muscle adults. You can see at the D figure with mark D. So we see with here we have a white arrow with basis. So, and this size around above 1 up to almost 1 centimeter. But then with the less than 1 millimeter, so most of them attached to the palm, coconut, and polypropylene or plastic fibers, I mean, sorry. So with different size, actually, we assume that the mussel larva attached to the palm, coconut, and plastic fibers, they age about 1 or 2 weeks. But then with the new cell attached to the mussel cells, they age about 1.5 weeks, 1 month and a half weeks actually. So we assume actually like because of if you see here, we assume that most of the larvae with the size less than 1 millimeter, they attach to the fibers. But then the new settler with the large size, about larger than 1 millimeter, actually they attach to the mussel cell. So I assume that this is because the pumping of the filter feeder, actually the mussel adults, might affect the attachment of the larva. It will be possible to be filtered by the adult. So that means the larva might refuse, came and attach to the mussel cell. So they might, the fibers actually in this case, with 3 kinds of fibers, become the favorite to attach on it. So the sides, we also, if you see here, I have 2 bars. 2 bars representative, 1 bar as side, Tiwoho and Blongko. And at the left side, I have the number of new settlers. So here the mean of new settle actually, where the figure show that the pattern of the mean new settle is higher in the Blongko than at the another side. It's called Tiwoho. But actually, this figure, it's probably more affected by we call by the time of exposure, especially when low tide. So at the Tiwoho is more time exposure, because when the low tide, the area that exposure to the sunshine, it's more time, because this is a large area when the low tide came. Dry, I mean. But then in Blongko, it has only short area that when low tide, it can be exposed to the sunshine. So that means when the because of this difference, exposure might hit the larva when they are already on their attached to the substrate, and it's possible many in Tiwoho might die compared to the Blongko. So we are also looking, like I mentioned, with the small aggregations, and then at the large parts we have the edge and middle of large parts. But for both sides, it seems that the pattern of higher new attachment on the small parts, it's higher than at the edge and middle of large parts for both sides actually. But here this figure might be affected by one I can mention, like cannibalisms. Actually, mussels actually can actually when they are filtered particles, the larva can be filtered by the adult of mussel. So I call this as is part of the cannibalisms. So, but when at the small pets, it seems to, when their new settle are higher, seem to be this position or these parts become a favorite for new settler to attach, but not at the large parts, whether at the edge or in the middle of patch. But then this is the type of substratum that I saw to you before. So we have palm fiber, and then we have coconut fiber, we have plastic fiber, and then mussel cell with biases thread. So I can show here the attachment vary actually, but from this figure, it seems that the attachment to the mussel cell with biases also seems to be at the lower positions. We did the statistics analysis actually. It showed that yes, the number of attachment is low, significant different with mass attachment to the mussel cell with biases compared with the other three substrates with palm, coconut, and plastic fiber actually. So from this figure, we can say that yes, fibers can be attract the larva to come and attach on the substrate actually. I would say this kind of substrate it's good for mussel larva to attach, but the mussel cell it's good substrate too, but because of the what is called the filter system, so make the larva try to postpone it to attach. But another things, possibility that the larva, when they came down to the bottom, when they tried to find a substrate, the good substrate, they might attach first to the palm, coconut, and plastic fibers attached, and but then when they grow for a larger size, here I can see more than 1 millimeter up to 1 centimeter. And then they probably can move and attach to the mussel cell with biases. I think it seems to me that this new setter get more confidence to attach after a primary attached to the different type of fibers. So the conclusion is yes, we have the effect of site, position, and position in the patch, and the effect of substratum on the settlement or new settle of tropical box mussel Septifer bilocularis. So considering the cannibalisms on predator, it seems that small patch is quite better to be attached by new settler rather than the large patch. So, and then the last one, this study, we suggest that the resettlement of box mussel on adult biases threads after first being attached to the substrate, palm and polyethylene, or I mean plastic substratum in the mussel bed is slightly occur, but then after that they can move to the mussel cell. So actually, this study can be used in my area. Actually, this because of the ecology function, like filter particle in the water. So usually the water can be clear. So it's probably the ecology function, like when the clear photosynthesis that's produced by, is running by what is called plants, micro and macro plants, will be good, and that's mean when the clear water, it's good for oxygen available in the water, and it will be good for the marine ecosystem. But however, usually our box mussel disappear in some part in our water, and also the population are reduced. So actually we are looking for how to replantations, the way of replantation this box mussel. So this kind of substrate will be important for us. So, and then this substrate will be not garbage, but we can use it to provide the mussel larva to attach. I think this is all for me, and thank you.